Hi, my name is Dennis Makau. I am a postdoctoral associate at the Department of Veterinary Population Medicine in the University of Minnesota. I would like to talk to you about porcine reproductive and respiratory syndrome, also known as PRRS. PRRS is a highly infectious swine disease caused by a single-strand RNA virus broadly classified as PRRS type 1 and type 2. PRRS type 2, which is predominantly circulating in the United States, has also been classified using two methods: restriction fragment length polymorphisms, commonly known as RFLPs, and lineages. There are 9 lineages of PRRS type 2 currently circulating in the United States. One of the more virulent viral strains, commonly known as RFLP type 174, also belongs to lineage classification L1 and predominantly constitutes the L1A sublineage of PRRS type 2 in the US. PRRS is associated with losses of over 6 million United States dollars every year due to piglet mortalities, reduced reproductive performance as a result of abortions in sows, and other management costs to try and mitigate the disease in the swine industry. As such, its management is of great importance to the swine industry. Our focus was to investigate factors that affect its spread in the US swine herds. To do this, we engaged 2,724 farms in a swine-dense area of about 26,000 miles here in the US. From these farms, we obtained animal movement data and all 5 sequences of all L1 viral isolates identified on those farms for the period of 2014 to 2017. Additionally, we divided the area into 13 grids and summarized on these grids vegetation cover, proportion of area covered by crop versus forest or trees, mean ambient temperature, wind speed, and precipitation, all of which are factors that have previously been shown to influence the spread of disease in an area. We used BEAST to analyze the phylogenetic distribution of PRRS in this study area. BEAST is a Bayesian analytical platform that uses the Monte Carlo Markov chain approach to allow us to make inferences of phylogenetic distribution and probability of genetic trees in our data. To enable us get to this point, we employed the general time reversible model, gamma distribution rate model, SkyGrid, and uncorrelated log normal relaxed clock model to allow us to infer phylodiversity points, time, and spatial diffusion of PRRS in our landscape. From our model, we observed that there were 4 main factors that influence L1 distribution in this area. And these factors were movement of animals, predominantly feeder pigs, the density of farms in a grid, the pig population in a given grid, and the spatial proximity of grids. Conventionally, if a factor has a Bayesian factor value of greater than 6, then that factor is considered significant and important in the phylogenetic diversity of your sequences. We further observed that the genetic diversity of L1 in this area had a seasonal appearance to it, peaking at around the months of May and June before decreasing and again peaking in the following year, and these are 2015, 2016, and 2016, 2017. In regards to the spatial diffusion network over the landscape, we observed that once the virus was introduced in the landscape, the virus spread very quickly over the entire area, and we had multiple introduction and reintroduction from grid to grid within the area within the 4-year study period. Ultimately, we conclude that phylogeographic diffusion of PRRS is influenced in this particular case by feeder pig movements, spatial proximity, and pig density. I would like to acknowledge all our funding partners, NSF, USDA, and NIFA, SHIC, and MSHIMP, who are our partners in data management and collation here in the University of Minnesota. Thank you very much.